It's my pleasure to introduce today's MBM Frontier Lecture speaker, Dr. Sepideh Sadaghiani. Dr. Sadaghiani earned her PhD in Neural and Behavioral Sciences at the Max Planck International Research School and is now an associate and is the principal investigator of the Connect Lab housed here at Beckman Institute. Over her career, Dr. Sadaghiani has won many prestigious awards, of which include her recent distinction as Helen Corley Pettit Scholar and a recipient of the Campus Distinguished Promotion Award. Dr. Sadaghiani's research is focused on understanding how the coordination of large-scale brain networks support flexible cognition with a variety of methods, including fMRI, EEG, concurrent EEG, fMRI, and intracranial EEG. And today she will be giving a talk on the functional connectome across temporal scales. And with that, let's give her a warm welcome.
Thank you so much for this very kind introduction and for having me. It's a pleasure to be here. I will be talking about the connectome today at its largest scale. So when I speak about the connectome, I mean connections between distant brain areas at some level of abstraction. And since I know that many of you are working at very different levels of neuroscience, I have quite a bit of introduction. If anything is too much or too little, just interrupt me and ask for further details so that I make sure everybody can follow along. Because I might not get to the end all the way, I wanted to start by thanking all the people that are actually doing the work. Here are the people in my lab that are working on this topic at the moment, and data will be shown mostly from Tom, my current postdoc, Jonathan, my former postdoc, and Paham, one of my senior graduate students. A lot of the data that I will be showing today are actually graciously shared with us from elsewhere, and I'm really thankful to that open access of the data. I'll be starting by a brief introduction of what I mean by intrinsic brain activity and intrinsic connectivity and what the connectome is at that scale. And then I will be talking about how we can use electrophysiological methodologies to measure the connectome, which is that still a very unusual approach compared to the dominance of functional MRI for measuring the functional connectome. The last two sections then focus on the spatial relationship between EEG-derived connectomes and fMRI-derived connectomes, as well as the temporal relationship between the two. So just to give an introduction for those of you who might not be as familiar, when I talk about intrinsic brain activity, I mean everything that is not extrinsic or evoked. Evoked brain activity is the majority of activity that we typically focus on in the cognitive neurosciences, which is a direct consequence of an external stimulus or an external event that we're interested in. Intrinsic or ongoing brain activity is essentially, as the name says, it's ongoing continuously. You might think of it as background brain activity, although it is very relevant to cognition, as I will show you in a little bit, and one particular study as an example. So now to talk about connectivity at this spatial scale and about connectomes, the way connectomes are typically measured, functional connectomes at this scale, are using functional MRI, and the brain is parceled into some parcellation or atlas of choice that might have different granularities and might either be based on anatomy or function or a combination of the two. But in any case, imagine you have separated all of your brain voxels into patches. And then you can, for any pair of brain regions, you can calculate a statistical measure of temporal relationship. So often it's just a linear measure. Pearson correlation is the most commonly used measure. And you can obtain a measure for every pair, and this particular pair shows a very high correlation in the temporal spontaneous fluctuations or intrinsic fluctuations of the ongoing brain activity. In contrast, these two other highlighted brain areas show a much lower correlation value. If you do that for all regions of the brain, you might actually obtain these intrinsic networks. In fact, at the bottom, these are derived using spatial independent component analysis, but there is other ways to get at these networks. But these distributed networks show brain regions across very distant areas that show particularly high correlation or other measure of statistical temporal dependency. And we think of that temporal dependency as connectivity, as some level of information exchange between these regions. So this is, for example, the auditory network. You can have posterior visual networks. You can have higher order networks. This is the dorsal attention or spatial attention, visual spatial attention system. And there's various other networks. This is one particular atlas that parcels the brain that way. If you plot the correlation values of all of the regions of your parcellation in a connectivity matrix like this, and then organize them by sets of regions that are particularly highly connected, meaning each one of these networks, you get this organization where you have the high correlations along the diagonal representing these canonical neurocognitive networks. But there's also, of course, relationships between those networks. So this is what we call a connectivity matrix. And one very well-established fact is that these connectivity matrices are incredibly stable across mental states, which breaks with tradition in cognitive neurosciences, at least the tradition that I grew up with. Your generation might be a little more elaborate, but this idea that somehow the brain functions in a reactive way. Something happens in the environment, or you have a goal, it might be an internal goal, there is some sort of a cognitive function, an event that is essentially generating this neural activity, or is a representation, an effect of this neural activity. But the fact that the connectome during tasks and during task-free states is very close to identical, essentially negates that understanding. So there is this organization of networks that I showed is incredibly stable across mental states. It has commonalities, very strong commonalities across people, even though there's individual fingerprints. You have your own network fingerprint that's really subjective to you, specific to you. But the point here in this slide is the stability of these networks. That said, in spite of the strong stability, there are very subtle but meaningful changes in the connectome between mental states, as well as from one moment to the next on a rapid time scale that do affect behavior. And one example is this study that we did. This is still fMRI, and we played a threshold level sound to the participant in the MRI scanner. And by threshold, I mean it was so faint that it was at the participant's detection threshold. So the participant about half of the time would miss the sound, and on the other half of the trials, the participant would hear the sound and report it with a button press. So the sounds were very, very far apart in order to be able to look at the unevoked activity of the brain that is free from these very slow and sluggish evoked responses of the fMRI signal to those stimuli. So the study focused on these areas where we have no evoked responses. And this is essentially that connectivity matrix that I showed you. Most of the time it's symmetrical, so that's why I'm just showing half of that here, and these are Pearson correlation values. But on every trial, the connectivity matrix, or between every trial rather, might look just a little bit different because we have dynamic connectome changes, reconfigurations. And when you plug these into multi-pattern classifiers, they can look at this before the sound is played, and they can predict, not with great accuracy, but certainly above chance accuracy, whether the subject is going to hear the sound or is going to miss the sound. Just based on the brain state many seconds before the sound even was played, I can look at the brain and say this participant is going to hear it this time. Same identical sound, or the participant might miss it this time. So there are subtle changes, but they are behaviorally significant. So that's a notion that really shows that the dynamics in the connectome are important for behavior. So the connectome, now not in brain space, just in graph space, these are the different parcels of the brain that I showed, and the lines are the connections between them. The different colors show those different networks that I mentioned before along the diagonal. of the connectivity matrix. This is just from one single subject. When I look at the graph of the connectome, right before the subject is going to hear it successfully, or before the subject is going to miss it. You can even just by just looking at it visually without running any graph analyses, you can tell that there is differences. All right, so this is more of a motivation for why I'm interested in looking at connectomes. Much of the rest of the talk is not going to directly link the connectome to behavior. I think there's a lot of basic work that first needs to be done, and I'll be mostly focusing on that today. So I think the way functional MRI-based cognitive neuroscience has developed is for a long time, we've been interested in finding brain areas that show high activation in response to some event of interest, tightly controlled for some other event of no interest. So, for example, if this study is interested in knowing whether and where there might exist a brain region that is more responsive to faces than to other interesting objects, we would show faces over and over again, many, many trials, and then we would make sure that our controlled stimulus shares everything with faces except being face-like. So it has to be an object, it has to be attention capturing, and must have similar features in terms of maybe we want to control for contrast or other aspects, image contrast. And when we tightly control that, then we look for the difference, and yes, then we find a single brain region. But that often makes us forget that no function in the brain is actually computed by a single brain region. If you actually just look at the main effect of your task, even the absolutely simplest task, even the absence of task, but just stimulation, you have a participant that's conscious and awake, and the MR scanner, if you show a picture or play a sound, you're going to see many brain areas that respond to that compared to the absence of any stimulus. It's that when we do those tight comparisons, we forget how much brain activity there is. So this study is just one example of the dorsal attention system I mentioned earlier, where the participant's attention is covertly shifted by a cue to one side or the other side of the space. And these distributed areas of the brain, parietal cortex, frontal cortex, the frontal eye fields, and the parietal sulcus, motion sensitive areas, they all respond as a network, in conjunction, in a correlated manner. What's interesting then is the discovery that this very network actually also exists when I don't ask you to pay spatial attention or any attention or do anything at all. If I have you lie in the scanner, even with eyes closed, even when you're asleep, I'm going to see that network. And it's not just that network, it's all the networks I was showing, right? And so we understand now that brain activity is distributed, this coordination is ongoing and continuous. We know that we can now bring in methods such as graph network neuroscience, network science that has been developed in other fields, and we bring it to neurosciences. And so that opens a new toolbox. I talked about dynamics, the fact that you can generate these networks and their organization across the whole brain parcellations over sliding time windows of some sort to try and see how they change in subtle manner over time. And finally, we've come full circle to say that these subtle changes in the connectome from one moment to the other actually affect the shape of this evoked response. And at the end of the day, they affect behavioral output, they affect perception, they affect all kinds of other cognitive functions. We've repeated a similar set of similar stimuli, similar experiments, similar to the one that I showed you, the auditory detection one, but using various different kinds of tasks from very highly cognitively demanding to very simple. And in all of those cases, we can show using machine learning that these state changes actually affect the evoked response and actually affect the behavioral output. However, the big problem is all of this is still in fMRI. So the fMRI signal, by its very nature that it depends on neurovascular coupling, is simply really, really slow. And we know that most cognitive functions don't unfold at that speed. And so this leaves us wondering whether we can observe connectomes and connectome dynamics with more real-time methods. So let me bring in one example here. Cognition doesn't happen at this infraslow speeds of fMRI. In fact, it doesn't unfold at any one single speed. We have various different cognitive, and by cognitive, I mean very broad, even simple perception of lines or sounds for me in this bigger picture are cognitive. Anything behaviorally relevant unfolds over various time scales, very, very fast and very slow and everything in between, and does so at the same time. We don't do something rapid and then stop. So the brain doesn't do rapid processing, then does something slow and then does something else. We do all of these things at the same time. So audition is a good example to visualize what I mean by cognition unfolding at numerous speeds and not just at fast or slow speeds. So this is essentially the waves, the auditory waves generated for a section of a nursery rhyme. And this particular paper actually broke down the modulations, the amplitude modulations of these sounds, or this the song rather, into the phonemes, the syllabus level, and the prosodic stress level. Okay, so the melody of speech. And you see that they're in very different frequency ranges at very different speeds. And it's not like we do just one thing or the other. The brain is operating at multiple speeds all the time. This holds for vision, this holds for all other aspects of behavior and cognition. So if that's true, okay, and I forgot. So the syntax, right, and the semantics are even slower than this. And then you might even read a whole paragraph in a book and you have that whole memory of the whole like logic and the connection of the story that you still have reverberating in your mind. And all of these things are still there at the same time as you're processing the rapid phonemes. So how do we go about studying the connectome if it is indeed representation of this vast intrinsic ocean of the brain, the majority of brain activity? How do we go about studying it actually at these more rapid speeds? So this just summarizes what I said. fMRI-derived connectivity is limited to the infraslow time scale, but cognitively and behaviorally relevant neural processes operate concurrently at numerous time scales. So I'm dedicating an entire section to electrophysiology and focusing just on time averaged connectomes for now, not the dynamics, because the majority of my colleagues in the cognitive neurosciences, if I went to a conference, the majority of people, if we asked them, they would probably still say, I'm not sure, you're really looking at signal or just noise. It's not like what I'm going to show you is accepted by most people, that you can do large scale connectomics using electrophysiology, at least non-invasive electrophysiology. But let me try to convince you that it's possible. Before I get there though, there is some caution to be expressed about the fact that electrophysiological measures are much more rich, in some ways, much more rich than fMRI is, because when we talk about connectivity in fMRI, we typically talk about some measure of statistical dependency over time, typically linear. We can do mutual information or other things, but we don't typically decompose the Bolt signal any further. Whereas when we come to electrophysiology, at least if we're thinking about oscillatory bands of electrophysiology, connectivity can either be defined in terms of relationships of the phase. So at the top, you see two regions here in red and blue, where the phase of a given oscillation is aligned between the two regions. So they're optimally aligning that between each other to have optimal information exchange, sending and receiving in a well-timed manner. However, alternatively, the amplitude of the activity in the two regions could be aligned, or we could have both. We could have phase and amplitude coupling. And there is second order coupling. There is amplitude to phase coupling within and across regions and so on. I'm not even going to get to that today. And in the following, I'm not going to make a distinction between phase and amplitude coupling. If for any study you're interested, I can say what we did in that particular study. Typically, we report both. And the other complication comes from the richness in the temporal scale. So EEG, if the amplifier allows, and it's not, usually the amplifiers don't record infraslow range, but you can set it such that it would record it physically. EEG can record the infraslow range of speeds. It can go all the way up to high gamma or high frequency activity. So we're crossing 4 orders of magnitude here, and it's very rich. It's also making the signal ways more complicated. We're not going to end up with one electrophysiological connectome, right? We're going to have many electrophysiological connectomes. In fact, that is really my point. I hope by the end of the talk, I can convince you that that really is probably what matters for the different speeds of cognition. Okay, so how do we do this? Most often we're interested in non-invasive signals. So imagine you get your channel-wise EEG signal. You have a complex signal. By complex, I mean multiple frequencies overlaid signal in every electrode, and then you might decide to filter it in different frequency ranges, although not always. Some studies don't. You definitely want to source localize your signal. So we often source localize to a dense mesh of a few thousand data points. And then we average those data points to the parcels of whatever atlas we chose. Every parcel will now have a band filtered signal of a certain frequency band. And we can choose to look at the phase or at the analytic amplitude of that signal. Then for every pair of brain regions, we can then construct a connectivity matrix, either based on pairwise relationships of the phases or of the amplitudes or some other higher order measure if they want to. But it's really important to take care of leakage, source leakage, and I'm happy to talk about that. But the major reason that I said my colleagues would express skepticism even after all the papers that have been published in this realm is that source localization is just naturally, by default, by its very nature, it's ill-posed. There is no correct one single mathematical solution. And the reason is that from the sensor level, almost unlimited, right, combination of sources in the brain could in principle generate the observed signals at the channel level. And for EEG, it's probably even worse than MEG because electricity flows over the skull, and so we have actually electrical leakage. But source leakage is a problem for both MEG and EEG. So we have some methods that allow us to mitigate this problem to a sufficient degree. It's not great. And unfortunately, it also throws away quite a lot of good signal. There is much evidence that this is an over-conservative correction, but there is no way around it at the moment. All right, so let's say we now have connectomes generated from EEG channels. What do they look like? So this, I showed you those networks first in introduction using spatial independent component analysis. This particular paper by Brooks and colleagues shows those fMRI-based networks in the top row. So these top rows of every subplot are fMRI networks. Okay, so you can recognize different canonical networks. You will get these networks no matter what, if it's resting state, if you're doing a task, if you're sleeping, these networks are there. They're intrinsic to everyone's brain, and they are essentially our internal architecture. What's astonishing about this study is they ran temporal IC on MEG, on source space MEG, just as I just showed you. They're generating source space signals. You can get very, very similar networks from MEG. And so that's essentially a proof, a side-by-side proof that yes, there is enough information in there. I'll show you more direct comparisons of the connectomes between, like quantitative comparisons as opposed to just visual ones in a moment. But before that, let me take you to intracranial EEG. So this data set is publicly available, and we're really thankful for that data set being shared by Kai Miller and colleagues. So these electrocorticographic measurements, so these are electrodes that are sitting invasively directly on the cortex, are in patients that are suffering from epilepsy, and they're being monitored for surgery, for surgical removal of certain brain areas that might be the onset zone of their epilepsy. Before surgeons do that, they observe these patients for many, many days in the hospital with these invasive electrodes, just to learn more about that patient. And so in the meantime, data can be recorded. And so I will show you connectivity matrices from patients like this, but remember that for every given patient, these are three different patient as examples, we will only have one spatial window into one part of the connectome. But typically they stretch over various functional regions, even different lobes of the brain. So to the left here, you see one single subject example connectivity matrix. These are electrodes, okay? So these are individual electrodes that were available in that patient in resting state and a very cognitively demanding two-back working memory task. What you can see here is in the theta band, for example, that the connectivity matrix averaged over the two-back task in resting state are quite similar to each other. And this is even stronger in the beta band. When we do this for all participants, I'm showing that here in the radar plot, it's actually pretty astonishing. So here I'm showing the dissimilarity, so that's the one minus the correlation. But what I want to highlight is if we split the task-free resting state in half and just look at the two halves of the data, how similar would these networks be if both were derived from task-free resting state versus resting versus task, and one task versus the other. And these tasks are very different. So this could be working memory tasks, language tasks, finger tapping. They all seem in the same range. It's not that task and rest are any more dissimilar than resting state is simply to resting state. So these networks are really stable over mental states, even measured invasively, when we don't need to worry about this source leakage and source reconstruction issues. Also, I mentioned different frequency bands. The organization, the intrinsic organization of the brain is highly stable over the different frequency bands as well. And it's also very stable between phase coupling and amplitude coupling. I just mentioned that for completeness. So what we've seen in fMRI in terms of an intrinsic organization existing in the brain and being very stable, the same holds true for electrophysiological measures in scalp EEG, MEG, as well as intracranial EEG. So just a summary for what I said up to now. The functional connectome is behaviorally significant. It can be studied with electrophysiological methods, even non-invasive methods, and it's very highly stable over different mental states. It's essentially our intrinsic architecture of ongoing brain activity. Okay, so do I have any questions here? Okay.
Yeah, Sepideh, maybe a naive question. Do you hear me?
Yeah.
Okay,
Go ahead.
so you mentioned about the stability of the connectome or the correlation matrix. Mm-hmm. Is it originating from the synaptic architecture, which is what I mean by that is once the neural network is formed in the brain, the pattern by which one segment is communicating with the other segment, that becomes stabilized. So
yeah, the answer is yes. The simple answer is yes. So generative models take, there's many examples, a lot of publications that take a diffusion weighted imaging derived tractography connectome, a structural connectome, or they even take tracer based connectomes for macaques. So that's much more precise than non-invasive tractography. In either case, you take a structural connectome, that anatomical backbone, and then what they simulate on top of that, just by computer simulation, is some sort of electrophysiological activity. Okay, so they might add some sort of noise in oscillators of the different brain regions, and just take the actual structural wiring as being the connectivity between them. And that model in itself generates the functional connectome. So we can show that the structural connectome in itself, by a simple electrophysiological simulation, results in infrasol fluctuations in the range of the Bolt signal that have a correlation structure the way the real functional connectome from fMRI has. Okay. So the answer to that is yes. And I think Joanna Cabral has done this also for the MEG-based connectome. You can derive that one too.
Okay.
So yes, it is, and that is one reason why I think that the MEG-AG-based connectomes are to some degree, and I will show you quantifications now, to some degree similar to the fMRI-based connectome, is that they have the same highway system.
Okay.
The same structural, right. And then there is the functional connectome, however, is not fully bound by structure, right? It's much more dynamic. Otherwise, we would not have any cognition, right? We would just get stuck with one single way of communicating between brain regions. And that is just not what we know the brain is doing, right? The functional connectivity uses those highways, but it can modulate itself to navigate that possible space of using those connections. That makes sense.
And is that where the temporal, the range that you showed, 1 to 100 hertz, plays a role? Like, there is a structural connectome, the highway system, but the connectome, which should be dynamic, is the dynamics coming from the wide range of frequencies that you just showed? Or is it?
It could be. So some of the data I have towards the end suggests that that might be the case. I just want to caution and say, when I showed the frequency bands, I showed them as pure oscillations. And we actually no longer believe they really are oscillations. They're just parts of the signal at a certain speed. They don't necessarily have to be periodic. Okay. Some of it might be periodic. You clearly are going to have a posterior alpha rhythm, for example, right? But yes, so what the talk is going to end on claiming that those different time scales are actually the different time scale of communication in the brain that cognition needs, because cognition is fundamentally multi-time scale. Yeah, and I could stop talking now, and so because that was the conclusion.
We have a question in chat from Miles Norsworthy. He asks, you mentioned that to a small degree, there is a connectome fingerprint unique to individuals. Does this fingerprint change over time? And if so, to what degree? And are there certain behaviors that drive it?
Drive the changes or result from the changes, I suppose.
Yeah,
very exciting question. So I'm not looking at individual differences of the static connectome. So the so-called fingerprinting field of connectomics often looks at this time averaged connectome. And it is possible for machine learning algorithms to pick you out among a lot of subjects, you as an individual, just by your connectome. And that's why they call it fingerprinting. It's really unique to you. In terms of how it changes over the lifespan, that's a very good question. I'm not deep in that literature, but the connectome itself, definitely there is changes over the lifespan that are consistent over people. And that has been extensively studied both for development as well as aging. In terms of the changes that are individually specific and how much they predict cognitive outcomes, for example, in aging, some people might have a trajectory where they show more decline than others. I'm sure people are looking at that. I'm just not aware of the specifics of that. In terms of individual differences, Marta, I know you have a question, so let me just do one more point here. We are interested in individual differences of dynamics. So remember those different connectivity matrices that change a little bit from one moment to the next and affect cognition? So we have a study published earlier this year where we looked at the heritability in twins. Monozygotic twins, genetically identical, dizygotic twins, non-twin siblings, and unrelated participant pairs in a very large data set. And we can show that the way the connectome changes over time from one moment to the next, so the particular trajectory of the dynamic changes of the connectome are incredibly heritable. And that means they're actually also fingerprinty-like, right? They are specific to individuals because they're genetically influenced. And so we are definitely more interested in more individualization of the dynamics, but that's just what my lab focuses on. Martha.
Well, I was going to ask a question similar to Tahir's, but now I've had time to think about it more. So when we talk about like one of the connectomes between two matching regions, isn't it correct to say that underlying that are the activities of individual neurons, clusters of neurons, megacluster of neurons that have their own characteristics, and this is the emergent pattern from the neurons that are making those connections?
I would say yes. It brings your question involves some aspect of spatial scale as well, though, right? So when I'm talking about these gigantic patches, that's why I emphasized them at the beginning, because I know that many of you don't work at that smoothed out level, right? we try to choose them in a meaningful way that they are somehow functionally homogeneous, right? So depending on how we choose the particular parcellation atlas, but they're somewhat homogeneous, right? They're not very homogeneous, and still they're gigantic. Even if we took there is voxel to voxel connectomes as well. People have given that up to a large degree because it hasn't been really useful. They're just computationally unbelievably big, right? You start with 100,000 connectomes and then you build the matrix of that. I mean, and how meaningful is that anyway? But even within a one MRI voxel, if we were to do voxel-wise connectomics, you would have smoothed out over everybody and everything, right? And for electrophysiology, it's the same. Even the invasive LFPs that we're measuring in those subdural electrodes, they are coming from large populations of similarly aligned pyramidal neurons, right? Or that's what we think at the moment is the cause for them, because otherwise we wouldn't see those neuropopulations. They have to be large groups that behave the same way. But yes, I think that what we're seeing in fMRI or electrophysiological connectomes are a sum of everything that's underlying it, assuming it's behaving sufficiently similarly over whatever abstraction level we're looking at to be able to see it. Does that answer your question?
Thank you.
All right, so let me quickly move on and say, I wanted to show you a direct quantitative similarity between EEG and fMRI-derived connectomes. I'm going to jump over that analogy and then go straight to this. So I just showed you how these EEG-based connectomes might be built in the different matrices, different frequency bands. Okay, so in this case, we would have one time averaged connectome per canonical EEG frequency band and one for fMRI. This is from concurrent data. Okay, so we record this data at the same time, the EEG and fMRI. And then we essentially source localize to the same atlas. So now the individual data points or edges or region pairs of these networks between the different modalities directly correspond to each other. So this is an early study that Jonathan Biesich did before joining my lab. Every data point in this plot is the strength of connectivity between one pair of regions, one region pair, but it's average across all the participants of the experiment, okay? So this is the population averaged connectome. And what this shows is for one particular EEG frequency band and one particular connectivity measure, which is a phase-based measure, the beta band, he's just plotting in, the paper has all bands, and so if you're interested, but this is just an example. He's plotting the strength of connectivity of that edge or region pair in EEG and in fMRI. And you see a moderate level correlation here. Now, what's interesting for me, and the first time I considered doing EEG-based connectomes, because I categorically ignored it up until that point, was when he showed me that if you actually draw the relationship between fMRI-derived connectivity strength and structural connectivity, and you do the same thing for EEG versus structural connectivity, the EEG, depending on your parcellation atlas, so caveat, within this data set is actually matching or outperforming the relationship between diffusion, structural MRI based connectomes and fMRI based connectomes. The EEG based connectomes is more similar to structural fiber tracks than the fMRI connectome is. And so this relationship is negative just because of the specific path length based measure of structural connectivity. So you can just imagine the direction doesn't matter, it's the strength of the relationship. So here, so more structural connectivity is actually linked to more fMRI connectivity. It's just the measure is derived this way. So what's interesting is that historically, when people first observed those fMRI-based networks, many were concerned that it is related to artifacts such as co-plumbing. Okay, so we know fMRI is based on the Bolt signal. And so if distant regions may, the way they receive blood, if it's somehow related due to the vasculature, then they would show correlated Bolt signal, but that wouldn't be related to neural activity. So people had many concerns. It could be head motion, all kinds of other things that affect fMRI signal. but they really felt good about finding this relationship. Okay, so in the mid 2000s, 2007, where most of the papers came out, to show that relationship and calm the field down and say, no, this is real, because it's actually related to structure. Well, this is better related to structure. So let's just embrace it. Let's do it. It's cheaper. It's not great, but it's working. So what I think about the effect size, though, I really want to emphasize is that Jonathan went ahead and combined a large number of data sets that were concurrently recorded. So it's 1.5 Tesla with 64 channel EEG, 3 Tesla with 64, 3 Tesla, 7 Tesla. So in all of these different data sets, the relationship, the spatial relationship between that connectivity matrix and fMRI and EEG is always around the same range. It doesn't matter what we do, even when he combines all of the subjects, right? And has a lot of subjects. If you combine all of these data sets, it just does not seem to pass that. There's the ceiling we can't overcome. So of course, this could be methodological, and I'm certain a lot of shortcomings of both methods are contributing to that ceiling, that we just can't make them be more similar to each other. But I want to actually propose something else, namely that there's neural reasons why there is not a stronger relationship. So just before I move to show you those reasons, I just wanted to clarify that in an intracranial study that built a single intracranial electrocorticography connectome by merging all of the electrode pairs that were available in over 90 patients. So if you put all the patients on one brain, okay, all the electrodes, you actually get almost full connectome coverage. Compare that to the bold functional connectivity connectome measured in a different set of people and averaged. This is essentially the same plot as I showed you before. Every data point is one edge averaged across people. Okay, the strength of one edge in electrophysiology measured invasively versus bold. It just doesn't matter what you do, even in the absence of source leakage. You are just capped at this 0.3 to something, just doesn't seem to be breaking out of that range. So let me tell you about, I'm just going to jump over that summary. Let me tell you about the temporal relationship now, because I think this really gave us insights into why there is a spatial relationship, but it is not perfect between electrophysiology and fMRI. I think time will tell us a lot more. So I'm now going to look at codynamics, meaning how the connectome rearranges itself from one moment to the next when we record the data concurrently. So we can look at the rearrangement in EEG and in fMRI at the same time. So the real question here is fMRI looking at the same neural process as EEG? Is it just the overall sum of it and then low pass filtered in time? Or is it measuring something entirely different? So I told you about the complexity of the EEG signal, that it has these various different bands and these various different ways of connectivity and so on. So there is a lot, when you look at the real direct electrophysiology, there's a lot of different neural processes that are going on. But at the end of the day, the cumulative energy consumption of all of those processes together is what the both signals should be able to see, right? So it's sort of the colorblind sum of all of these processes, but then squeezed through the low pass, temporal low pass properties of neurovascular coupling. So is that the case, or are we looking at something slightly different? So I showed you this one before. The only thing that we're doing different here is that instead of having a single connectivity matrix for one given EEG frequency, we now have a windowed connectivity over time for one given frequency of EEG, and we can have that for every frequency band. And we do the same thing for fMRI. So now think about a time-resolved connectome, okay? And we do that in both methods just to see how the changes in the connectome over time may relate between the two data sets. So this is just a hypothesis figure of what we might find. In principle, one could imagine that EEG here in blue and fMRI in red are expressing different connectome patterns. So these are just cartoons of graphs. that the brain might switch between, okay? And so let's imagine there was a common space of possible connectome states, okay? So the space is shared, and EEG and fMRI can visit those same states, can become similar in spatial arrangement. That would be here, or we could imagine that the brain states that we can see in EEG have a spatially entirely different arrangement than those that we can see in fMRI. That's another possibility, because now we're in the dynamic range. It doesn't necessarily have to align with what we saw in the static domain that I just showed you before, static or time average. We could also imagine that the states are spatially similar, so we have somehow a mingled state space, but that they are actually not temporally aligned. So when EEG is in this state, okay, that might happen at a different time than when fMRI is in that state. Now, how could that be possible? That could only happen if EEG and fMRI measured different neural populations. Otherwise, this scenario can't really occur, that you would have somehow a different sequence of states in one modality versus the other. Do these hypothetical scenarios kind of make sense? Yeah, all right, okay. And so I'm now coming to a data set that's really unique in the world. This is data, again, very graciously shared with us. This is from London, has been collected over many, many years. This is intracranial EEG, both depth electrodes and subdural corticography electrodes under the skull, at the same time as fMRI. So these are the same type of surgical patients. They have electrodes in their brain and they've been put inside the MRI at the same time. So this is the best scenario, right? Unless you can do animals, right? And have the luxury, as Hussein has, to actually do these things in animals. In humans, this is really, you know, they had to do a lot of safety studies to make sure that they can do this. So it's a very unique data set, but it's essentially the perfect one where we have electrophysiology undisturbed by source leakage, of course, covering only one part of the brain. But then we have fMRI at the same time, so we can look at the fMRI close to those same electrodes at the same time. So we wanted to first establish some of these effects in intracranial EEG with concurrent fMRI, and then we replicated in healthy participants with scalp EEG and fMRI. So let me show you what we did. We're calculating really rapid windows here. And so this is the fMRI signal relationships for all regions over all the electrodes, close to the electrodes. So this is the connectivity matrix. And then we have one for one frequency band. I don't know which one it was, can't remember. For one particular frequency band, we have amplitude coupling of that frequency between the electrode pairs and phase coupling. And so for phase coupling versus fMRI, let's say, between these two matrices, at every time frame, we can generate the spatial correlation between these two, the spatial similarity. And I'm simply plotting that spatial correlation over time here. So the spatial correlation goes up and down. It becomes similar, more similar, and less similar. And we then compare that to a null model. How likely would it be that by chance they would look this similar? Okay. So on this other side of the screen, you see the time periods in which the similarity for any given frequency band of the electrophysiology significantly surpassed chance, the similarity to fMRI. And we found about 20% of the time frames. Now that exact percentage depends on a lot of choices and of the data. It could go up and down, but it's small to moderate in terms of the overall occurrence of similarities. The other thing that we also observed is that the frequency bands don't all become similar to fMRI at the same time. They switch back and forth to a large degree. Now, the next graph, and bear with me, I'm soon done. This one looks complicated, but I'll break it down for you. Just ignore these parts and just focus on this matrix. Every data point in this matrix is a spatial correlation value between the connectivity matrix of fMRI and of EEG. That's the video curve I just showed you, okay? So that's spatial correlation, the spatial similarity. On the diagonal is real time. So the time in fMRI and EEG correspond to each other here. But off the diagonal, we have time running from the first point of data collection to the last point for fMRI here, and for EEG on the other axis. So we actually slide the data set against each other and recalculate the spatial similarity every time. Now, every time we surpass the null model for one given frequency band, we put a dot in this matrix. This is, we then correct for a lot of multiple comparisons. But what's important about this whole graph, even if the details are not clear, is that the spatial similarity between EEG and fMRI occurs as often in sliding lagged time as it does in real time. So we don't think they're actually measuring the same connectome arrangement, just with different goggles. Okay, I have the neurovascular goggle and I have the electrophysiological goggle. We actually think they're looking at different time scales of the connectome. And so let me come to this, and I make this my last slide. I'm going to skip the next two slides. The scenarios that I pointed out, we think that the middle scenario is closest to our data. So the state space, the arrangements of the connectome that we can observe in EEG and fMRI are somewhat shared. Okay, they're shared above chance. There's some level of spatial convergence. But which states are occurring at any given time and how they are switching to any other state and moving through the state space is independent in fMRI in any one of these EEG frequencies. In fact, if you looked at one single time point randomly chose time point T10, okay, take that single time point here, fMRI would be in one brain state and one graph configuration, whereas each band of electrophysiology would be in a different configuration. So we have a multiplex of networks at every given single time. So this region and this region do not have to, so this region can be talking to this region in one channel at one speed, and at the same time, it's having a different conversation with a different set of brain regions using a different connectivity mechanism and a different connectivity speed. So we can have so we're talking about the functional connectome. I don't think we have the functional connectome. We have the functional multiplex. And this multiplex is a multiplex of time scales. And all of it is dynamic, and they're all operating in synchrony. I'm gonna leave out some spatial and temporal rules that we have found about that space that I just showed you. I'm just going to jump over that. I'm just going to go directly to the conclusion now. fMRI and EEG-derived connectome dynamics comprise spatially similar states. The state sequences are somewhat similar. I just jumped over that, but take my word for it. But the speeds are not the same. So they share relatively similar trajectories. They just don't unfold at the same speeds. And I really like to close by citing Rita Hari. And for those of you who did hear her talk or did not, and I just shrunk the URL. You can also just type it and you'll find it online. It's a keynote lecture from HBM in Rome in 2019. She writes, MEG-EEG weighs strongly the fastest conducting pathways, whereas fMRI probably receives its main contributions from neural ensembles that are connected via slow and thin fibers, thereby apparently reflecting functionally different brain activations. We don't think about fMRI that way. We just think it's an epiphenomenon of whatever electrophysiology is measuring. But in this talk, she shows data set after data set that we're trying to somehow bring together what they measured in MEG and fMRI, and it would just not align, because they might actually be both meaningfully recording different aspects of brain function. And the brain is a multi-time scaled system, she claims, because cognitive processes unfold at different speeds. And I think the data I presented shows that this extends to whole brain connectome organization, and that creates this multiplex of connectome dynamics over time. So the next big challenge will be to connect these timescales directly to timescales of cognition to prove that this theory is actually true, right? So the connectome measured at different speeds should be affecting different aspects of cognition, depending on how fast or slow those aspects are unfolding. And as you might imagine, we're working on conceiving experiments that can do that, but it's quite challenging. And so with that, I close. Thank you. Thank you so much. All right, let's see. And are you keeping track of whether there's anything in the chat or?
Yes, there is a question from Jonathan Cerna. Oh, sorry, no, not a question. Sorry.
Okay, hi, Jonathan. Okay, I just put some pretty bold claims out there. No one's resisting.
How long do you think it's gonna take to solve the riddle you asked Davis?
Oh my goodness. I definitely think having multimodal data helps. But perhaps we would start with the different timescales of EEG offline. I'm not sure. We have some data collection ongoing. It's been ongoing for a few years now. It's a large scale grant that we're recording over 100 subjects, 3 sessions concurrent. 2 of the sessions are concurrent imaging. And the tasks are designed for another question, but the participants get on every trial, they get a trial from one of 3 possible experiments randomly chosen, so they can't really prepare what they need to do. And those experiments are very different from each other. One is simple signal detection. That's the experiment I showed you, is just a beep. Did you hear it or did you miss it? The other ones are super complicated, cognitively demanding working memory tasks. And there is something in between, which is emotion valence judgments on ambiguous images. So we have a range of different cognitive processes, and we have concurrent data. So even though that's not the main goal of that data set, we could look whether the connectome dynamics in EEG and fMRI are affecting those different tasks differentially. Because some of the tasks can be processed really rapidly. There's just a sound and you just press. The other one is, okay, look at this display of 8 possible arrows. Then they disappear. You have to keep them in mind. And a long time later, we'll probe you on one of those and which direction was the arrow point. So that's a much more complicated function. I don't know if complicated translates into slower. So we have to think about whether we can use that data set. We have designed a new task that we haven't started collecting yet that should get a little bit more directly at these timescales. Yeah, I think it's years away, I think, unless someone has a perfect data set or some idea. I mean, maybe something like, I'm sure there is data sets around, at least with EEG, looking at naturalistic speech or songs, right? And if we can parse out the aspects that I showed you, phoneme level, syllable level, prosody, word, syntax, and semantics, we should have a whole range of timescales. So if we have EEG with enough channels that allows us to source localize to a whole brain connectome, we should be able to at least answer some of that with existing data. So yeah, getting at the infra slow range, that's going to take longer because then we need the concurrent data.
Yeah, if you talk about these multiple timescales, how do they map onto the traditional frequency bands or
the timescales of the cognitive functions?
Yeah.
Or that is a good question. I put in only one example, and that wasn't even from my own expertise. I know nothing about language, okay? And this is clearly in language, but if you go to in this particular song, which also had a certain speed that doesn't necessarily need to match if the words were spoken, right? But here you see like basically delta range. This is, yeah, essentially low theta to alpha, and you'll have upper alpha to beta range, right? But is that really going to translate to beta connectomes affecting phoneme perception versus, you know, alpha range affecting whatever syllable level perception? I don't know that. So, and also, how do I probe that concurrently? I mean, that's hard. I would have to probe each one of these cognitive functions separately, right? Whereas they don't really happen separately from each other. It's really hard, yeah.
Yeah, because these bands, they've been determined like 100 years ago or whatever, and everybody sticks to them. How accurate
Yes.
is that now?
So I can tell you that when we generate these connectome streams, and the slides I jumped over, I can just pull that up because it's nice to talk about that. These are just two of the canonical EEG bands, and this is concurrent fMRI over 6 seconds. And the color code here is brain states. Okay, they spatially correspond to the colors in the bands, but you see that the faster connectome speeds are switching much more rapidly. And so this is really scaling that way. And however, if we don't cut these ranges by the canonical frequencies, we just arbitrarily cut them differently, we still see them arranged by the speed in which we filtered them. I actually no longer believe we're looking at oscillations. I'm just thinking we're taking one part of a signal that's a sum with a 1/f frequency spectrum over a whole lot of different electrophysiological functions. And whichever way I cut them, I get some. So what I want to say is I actually don't think we have 6 connectomes, 5 EEG bands, and then infraslow. I mean, I'm scared to say it, but there might be an infinite malleable, non-dissociable kind of gooey connectome complex that is not actual layers. I can't even visualize it in my mind how it is. I actually don't think these bands, when we cut them different ways, we get the same results. So I think our results are not bound to canonical frequency bands, which is scary, very scary. So I hope they're still true. We're trying to convince ourselves we're not measuring noise.
Sepideh, I have a question. This is very complex. So maybe a very naive question. So in one of the results that you showed that seems like there is something that makes this probably plus fMRI results that they're all the same. So it's like 0.37 or so that they're kind of capped.
Yeah.
What's the significance of it? I mean, what if they would not be capped? What if they would be very diverging? Yeah, that's the plot. Yeah.
So yeah, these two plots. So when I talk about static, including these plots, I'm talking about time averaged. Okay, so when you take you make a single connectivity measure over the entire recording, which is what we've been doing for much longer than dynamic connectomics. So the significance of that, in fact, I should mention, if you build a model that concurrently takes all of the EEG connectomes at the same time and takes all the phase-based connectomes and amplitude-based connectomes of EEG at the same time, all of it, it can actually predict fMRI at a 0.7 or so, rather than 0.3, 0.4 level of correlation. Okay, so you can improve it if you take electrophysiology a bit more holistically. But for individual bands, the significance and why I emphasize this is to end on the notion that I think fMRI in its own right is measuring a different type of neural population. Not fully, it's certainly overlapping with what we're seeing in electrophysiology, but I don't think I don't think neurovascular coupling takes us to just an epiphenomenon of electrophysiology. If what Rita Hari is saying is true, then fMRI is simply more sensitive to slower processes and sees them better than MEG or EEG can. So fMRI is not just the sum of what EEG, MEG can see and then pass it and make it low, temporally smooth. fMRI in its own right might be better in one particular part of neural activity. She goes so far as not to just say they are thin and slow fibers and unmyelinated. She also goes ahead and says they involve more modulatory neurotransmitters than direct fast neurotransmitter connections. She actually completely thinks these are really different neural populations or different neural processes, which is quite shocking. I remember the murmur that started in Rome once she was done with her talk. And I'm like, well, so this is a keynote lecture where there's 4,000 people sitting in this hall. And then she ends on, the talk is called Timing Matters. And, she's a giant. You're not, you may not have heard her name because, she's in cognitive neuroscience, especially MEG, she's, just this figure. And after all of these many decades, she summarizes essentially her insights as timing matters, and MEG and fMRI are not measuring the same timing in neural space. It's kind of it's a very bold statement, but I think this is the most compatible with our observations. Yep. Okay, awesome. I need to unfortunately jump off, but I thank you all.